Good evening everyone. Welcome back to AgriAddict. We are starting in 5 minutes this session. So please share this link with your friends who are preparing for ASRB. This session is very important because we came up with the most expected and important questions for ASRB. Hope everyone is preparing very well for ASRB because just 1 week left for exam. It is on 2nd, right? Don't be panic at the last moment. Only mock tests can help you for a revision. So, hello Disha. Good evening. Okay, shall we start our session? We start. Okay, let's start our session today. Good evening all. AgriAddict welcomes you back again, and I am your mentor Gayithri, MSc scholar from Division of Seeds and Technology, studying at Indian Agriculture Research Institute, New Delhi. So, in continuation to the previous part, that is part 2, we have uploaded just 2 days back. We came up with even more interesting and important questions as a part 3. So, let's start our session today. So, the first question is, the damaging stage for Capra beetle is? The options are grub, egg, larvae and adult. I'll repeat the question. Damaging stage for Capra beetle is? Try to answer this question. So, as we all know, this Capra beetle is a storage pest, right? It's a very important storage pest. It is a primary feeder and internal one. What is primary feeder or internal? Those pests which directly feeds on the seeds, right? And inside the seeds, like the germ part or endosperm part, they will directly feed on the seeds. They are primary feeders and internal feeders. So it is a primary internal feeder. And what is the damaging stage for Capra beetle? So here in this diagram, you can see this is a Capra beetle, which is a brown colored body with hairs, right? But the damaging stage for this beetle is larvae. It's also brown color with a cushion hairs at the posterior end of the larvae, right? So this larvae, the damaging stage in Capra beetle. How to remember this? Since you have so many storage pests which are very important, in that you have to remember which are primary, secondary, internal and external. We have come up with a one trick, that is KL Rai. You can remember in any way. KL Rahul also. Here, what is KL and what is Rai? Here K is Capra beetle, L is long floored beetle. These are the beetles, or we have so many beetles, right? As you know, saw-toothed beetle, that red floor beetles and all. Among those beetles, only these two beetles, the damaging stage is larvae. For other all, both larvae and adult. For all other beetles, the damaging stage is larvae and adults. But for these two, that is Capra beetle and long floor, this is only larvae. And if you come for moths, since you know the storage pest, if you come for a composition or proportion of causing, Coleopterans are in major, followed by Lepidoptera, followed by Hymenoptera. So in Lepidopterans, we will see the moths. So among these moths, 3 moths, that is rice moth and A for Anguimous grain moth, and I for Indian meal moth. These 3 moths having a damaging stage of larvae, and all the moths having a damaging stage as a larvae and adult. Just remember this. Since you can't remember for every pest, you have to remember for exception so that you can easily exclude the options in an exam. So, based on this, we can say that the damaging stage for Capra beetle is larvae. Right. So many people have answered it. Let's go to next question. So, the fumigation is not fully effective below at temperature of. Fumigation is not fully effective at what temperature? Options given are 15 degrees Celsius, minus 15 degrees Celsius, 20 degrees Celsius, and just make it 30 degrees Celsius. Okay? Options are 15, minus 15, 20, and 30. Answer it fast. At what temperature the fumigation is not effective or fully effective? Somebody answered it D. Okay, 30 degree, and others. Okay. Hope you all know what is fumigation, where we do it in storage to control insect pest, keep our seeds healthy, or keep our seeds free from attack of this insect pest. We will go for a fumigation which is usually done in a gas tight chamber, right? Why gas tight chambers only? Because fumigation means the name itself indicate we are doing in the form of fumes. Just to not make these fumes to get escape from the room, we are doing it in a gas tight chamber. But at what temperature it is effective? Why fumigation is usually done? For insect pest. To know this, you have to know the concept. So, insects, at what temperature they are highly active metabolically? At what temperature they are active? Usually above the 10 degree or 15 degree they are effective, and if it comes to moisture content, above 8% moisture, right? Above 8% moisture. That is why we are keeping a seed in a store at a low moisture content as much as possible. So if you see the options here, they have given 15, minus 15, 20, and 30. So with this concept, you can easily say that at 30, insects are active or not. At 30 degrees Celsius, are they insects active or not? Of course, they are active. At 20, they are active. At 15, they are active. What about minus 50? They are not effective, right? So metabolically, they are inactive. That is why we should not do fumigation at a low temperatures like minus 15, or in general below 10 degrees Celsius. The fumigation is not fully effective, since that fumes or the fumigation product will not go into the body of insect because it is metabolically inactive. And one more thing is, at this low temperature, that is minus 15, the product will get a low diffusion rates. It won't be diffused in a faster rate, so it will accumulate at a one place only in a storage. That is also a problem, because the fumigation, the rate or dosage of fumigants we are using, just if we see that if it is too low, that you can't even control this pest. If it is too high, it will immediately attack the seed vigor or viability. So just carefully you see the dosage. So this dosage, whatever we are using, if it is at -15, it won't be diffused in a uniform way, and the insects are also inactive. So the answer is -15. Hope you clear. Let's go to next question. So what is the seed multiplication ratio of groundnut? Very, very important topic for exam, where you will get a very direct questions on SMR, right? Seed multiplication ratios. So try to answer this. SMR of groundnut. See the options first. 1:20, 1:50, 1:8, and 1:4. You have to remember these things because these are factual. Okay, Akash answered 1:4, and others. Okay, first get into the concept. What is seed multiplication ratio? So the name itself, it indicates seed and multiplication. If you sow one seed, right? You have sown the one seed already, and you got around 40 to 50 seeds. Just assume. Vary with the crops, that is why the concept, right? You got the 40 to 50 seeds from one seed. Ratio means so that means you just divide it, that is 40 to 50 you got, right? That means that seed multiplication ratio of that crop for that particular crop is 1:. So once you sow the seed, how many seeds you harvested from that seed is a seed multiplication ratio. So the for groundnut, it is 1:8. Please note of this. Pratibha, right? Sharin, Disha. Yes, 1:8. It is for groundnut. And also look for other crops also, mainly for cereals, important pulses, and also oil seeds, because you will get a definite question from this SMR concept. Okay, and this is for potato. And try to remember those crops which are having a very, very less SMR also. Okay, am I clear? The next question is, power of seed inspectors comes under which section of Seed Act? The Seed Act means, as you all know, 1960. The options are section 13, 12, 15, and 14. Answer it. Pratibha, Sharin, Disha, Akash. Right, right. Shrikant, you answered it right. This is also factual question. Seed Act 1966. Just tell me how many sections we have in Seed Act. How many sections we have in Seed Act? Okay. There are 25 sections. And in 25 section, you have to remember all the 25 sections, because you can get question from any section, and these are easy also. To get PDFs of these, just fill a Google form, which we have provided in the description, so that you will get all the PDFs whatever we uploaded. We have given ISTA, IMSCS standards, and seed legislation PDFs, and all other important PDFs where you will get the direct concepts. So if you fill that Google form, you will get directly into your WhatsApp. Just check the description box later. So in this 25 sections, we have important sections like section 5, 6, 8, 11, 12, then 21, also the 16, okay, 9 and 10, where it is related to application, right? So here the question is power of seed inspectors. Yes, yes, people answered it very right. That is section 14. So many people will get confused because we have a 3 sections related to seed inspectors. from 13, 14 and 15. Remember these. 13 is appointment of inspector. Yes, this section will tell about who will become seed inspectors, what is the eligibility and all. 14 will specify the powers to the seed inspector. And 15 is the procedures to be followed by seed inspectors. Maybe the field inspection or during a sampling. Okay. Right. So remember these three sections 13, 14 and 15. 13 is appointment, 14 is powers and 15 is the procedures followed by the seed inspectors. So the option is D, section 14. Next question is seed rules. This is also seed legislation part. Seed rules contains how many parts and how many clauses. Options are 12 and 28, 11 and 28, 11 and 38, 12 and 38. If you see the options, you have to be so clear so that you can't even exclude if you are confused with the topics. So wherever you are getting a factual things, you have to mug up or you have to thoroughly read it so that you won't get confused in exam. Okay, answer it. How many parts and how many clauses in seed rules? Seed rules in which year? Is these rules will specify how to enact or how to function seed act, right? All these act functions and all are monitored by or according to these rules has to be functioned. So in seed rules we got 11 parts and 38 clauses. Right, Shrikant, Sherin and all answered it right. Pratibha, just look into it. It is 11 parts and 38 clauses. Okay. Try to remember at least the important provisions because if you know the seed act, you can easily remember the seed rules. The heading of 11 parts you have to remember. Okay. So the option is C. The next question. Validity period of seed dealer's license. Validity period of seed dealers 9 months, 6 months, 3 years and 5 years. Answer it. Validity period of dealer's license. First tell me where will you find this dealer's license concept? Seed dealer's license. In which act or in which legal provision we have this license? Somebody answered it 9 months. Okay, let's get into it. Mainly we are getting as a provision under seed control order in 83. Dealer's license, those persons who are involved in a seed marketing and all, right? And the validity period of this license is 5 years. Don't get confused, I'll explain you. Earlier it was 3 years and this seed control order is amended in 2019 and increased this validity period from 3 to 5 years. Remember this. You have to be updated with these provisions. You have to see some amendments also. Okay. So the answer is 5 years. Where you will get this 9 months and 6 months? People will get confused because 9 months the validity period of whatever the seed certification. In seed certification we are getting a tags or labels, right? So those seeds which are get certified by seed certification agency, that certificate is valid for 9 months. And if you want to get it revision, so that revised one is valid for 6 months. Okay. You see the conditions also when it will be reapplied and when they will give a revalidity to this. So it is a validity period for seed certification. Then it is a validity for seed dealer's license. Earlier it was 3 years, but now it is revised to 5 years. Hope you got this. The next question is rules for protection of plant variety and farmers' rights came in which year? It is 2001, 2003, 2006 and 2005. Options are like this. So tell me rules for the protection of plant variety and farmers' rights. PV and FR. Yes, yes, Shrikant, right. It can be revised for any number of times. Right, Akash, Farooq, 2001, and others. What do you think about this? The rules for PPV and FR came in which year? Here, right. It is in 2003. Just carefully read it. They have asked for the rules, and PPV-FR Act enacted in 2001, and rules came in 2003. And the PPFR authority is located at New Delhi, just here in a mass complex, right? People might be knowing this. Authority is located. Sorry, authority is established in 2005. And regulations, don't confuse with the rules and regulations. When they established authorities, they have given some regulations. So it is also in 2005. So remember this. Act enacted in 2001, rules in 2003, authority and regulations in 2005. Right, Raju? It's right, 2003. And let's get into next question now. It's Orange International Certificate is issued for. Orange International Certificate for seed lot size, submitted sample, and working sample. Options are like this. So answer it. Orange International Certificate will be issued for what? Is it for lot or lot size or samples? You had to answer it fast since it is only 1 week left. There are all the factual things. You have to remember each and everything, and these should be at your tip of the tongue, right? Okay. Okay, ma'am, when registration started? It is started in 2007. Raju, under PPFR Act, crops are started registering in 2007. Right, right. People are started answering it, and they answered it right. This is for seed lot. Okay, tell me what is Orange International Certificate? We have two international certificates under ISTA, right? Orange and blue. What are the difference? If we go for any certificate, that means it has got sampled and it has got tested. After getting sampling and tested, they will provide for certificate, including what are all the standards it has reached, right? So in Orange International Certificate, what happens? Sampling and testing. These both are done by ISTA accredited laboratory. Okay. If both of these things, like sampling and testing done by ISTA accredited laboratory, so that means it is related to seed lot, because the same laboratory has sampled it, the same laboratory has tested for this. So it is a given for seed lot. But in blue international certificate, what happens? Only the testing part. Okay. Only testing of that sample is done by ISTA accredited laboratory. Okay. Sampling may be done by any others. So it is not related to any samples, because they don't know, while testing they don't know how they sampled it and how this sample has obtained. Just whatever we have submitted, they just test it. So it is related to sample, only the sample, not all lot, because they don't know about the lot. Just whatever the working sample, submitted sample they got, they just tested for it. So it is a certificate for seed sample, and this is a certificate for seed lot. Don't get confused, it is very important. So just remember, orange is for seed lot, blue is for seed sample. Earlier we had a green, but now it is yes. Okay, just don't think about that. Right, Akash. It both are done by ISTA accredited laboratory. Okay, am I clear? So the next question is, in a typical 4-screen model, that is air screen cleaner, which function is performed by the third screen? The options are like this. Aspiration, scalping, close scalping, and grading. Answer this. Very, very important and very basic equipment. We discussed in the last part, right? Air screen cleaner. They have already mentioned it. So don't get confused. Four screen models they have. And which function is performed by the third one? But he answered. Okay. Right, Sherin, Farooq. Okay, you answered it very right. Close scalping. Okay, let's get into this concept. First, they have told that air screen cleaner, as we all know, it is a very basic equipment, right? First equipment where seed flows is air screen cleaner. It will come in a basic cleaning. So it is based on the seed size. Based on this, it will grade for. So the number of screens may vary based on the crop or based on the quality of a seed we want. Here they mentioned 4-screen model. In 4-screen, usually, don't think that the first, second, third, fourth, when we have 4 screens, don't think that first 2 will do scalping, then second, then third and fourth will do grading. No, this is not the condition. When we have a 4 screens, first is for scalping, second is for grading, and third is for close scalping, fourth is fine or close grading. If you see this, when we go for actually 2 screens are enough, right? In basic or in general, we'll just think for 2 screens. One is scalping for one, the other is for grading. But if we are going for more, increasing the number of screens, that means we want to get a very quality seed or pure seed. So in scalping, what happens? It will remove those materials or those trash materials which is having a size more than the crop seed, right? Crop seed means ours. So in first screen, all the crop seeds will be coming to the next screen, and what are all the trash materials like stones and all, they will be remaining and they will be disposed to the outlet. And in grading, what happens? The size of those materials which is less than our crop seed, like chaffy materials or any sticks or any weed seeds also, some, and some larvae of insects and all, which are having a size which is lower than the our crop seed, will be screened out. The next, after this also, if we want more efficient cleaning, then we go for a close scalping, where here we are having a perforations bit smaller than the first screen, because whatever the very big trashes, still if we have any left with any stones or any clods of soil, so we will go for a close scalping. The same way we will go for a fine grading. Okay, understood? So the third screen is for close scalping and fourth is for fine grading. Don't confuse with the grading and fine grading. Okay, grading is by second screen and fine grading is by fourth screen. Understood? So the next concept, what should be the temperature of drying air in a heated air drying? So options are 120 degrees Celsius, 110 degrees Celsius, 110 degree Fahrenheit and 32 degree Fahrenheit. They are asking what is the temperature of drying air in heated air drying. Since seed processing is a very important unit, you have to be thorough with all those, from drying, from all the processing steps, okay? Drying, grading, cleaning, packing, seed treatment, seed quality enhancement techniques, everything is under seed processing, and also the testing part. So here you explain me in drying, we have types, right? That is, one of the type is heated air drying. What is the temperature of it? Right? People are answering it 110 degree Fahrenheit. Okay, let's see it. Heated air drying comes under what type of drying? We have a natural air drying also, and we have a forced air drying, right? The name itself indicates forced air means we are forcing the air artificially. So that for drying, obviously we need some heat, right? The heated air. What is the principle behind the forced air drying? I'll explain in a simple term. It is seed water pressure. Water vapor pressure. And atmospheric RH or pressure. Okay. To see this, if we see the any gradient, usually it moves from higher to lower, right? So, if seed is having a more moisture content, more water vapor pressure means it is having a more moisture content than the surrounding one. Then what it happens? It start losing it, the moisture, right? If seed is having a very less, for example, it is 11% outside and our seed is having 8%. Then what it start? It start gaining the moisture content, which is usually undesirable, right? So based on this principle, usually for drying means we want this condition, right? We want our seeds to lose the moisture into the atmosphere. For that, we have providing the artificial air or from an outside source, from an external source. In that, we have a three types. That is natural air drying. Oh, people? Okay. I'll explain. These three types are natural air drying, air with supplemental heat. You have to remember this because just remembering these terms will give you the explanation. Supplemental heat and then heated air drying. Okay. In natural air means we are not using any extra for external heat to the air. We are just providing a natural air. In supplemental heat means we are giving just 10 degree Fahrenheit to 20 degree Fahrenheit so that the seeds will lose the moisture bit quickly. And in heated air drying, we are giving a 110 degree. Don't confuse with the degree Celsius and Fahrenheit. Just see the units, okay? 110 degree Fahrenheit heat, sorry, temperature. Okay. Okay. These two systems usually in India we won't follow it because it is too slow, right? But it is a quick method and it is usually conventional also because this is done in either in a bin or any storage structures we can do it. We have to just create a one building or any storage structure that we have to provide some duct system for air like air duct system and we can supply the heat continuously. So just see for a duct system also where we have a three different one, okay? And so in heated air drying we are providing a 110 degree Celsius, 110 degree Fahrenheit, sorry. Okay, am I clear? Right, Pratibha, Sharin, Chote Bhaiya, you are also right. And the next question is, International Seed Testing Association was found in which year? The very basic question everybody has to answer and very important question. Options are 1920, 1924, 1966 and 3. Answer it. International Seed Testing Association was founded in which year? Fast guys, you have to answer this question. Right. Akash, right? It is 1924. Yes. ISTA is an International Seed Testing Association founded in 1924. Just see the background. Like when they have started a proper seed testing or proper seed certification system they need, they started holding a congresses like annual, or you can think as an international congress, right? Congress means meeting. The first congress held at Hamburg, it is Germany, in 1906. It is the milestone. Start. Okay. The second one is also important, which held in 1910 at Wageningen, it is Netherlands. And the third congress is held in 1921 in Copenhagen, that is Denmark. It is important because European Seed Testing Association, which is later turned into ISTA, right? So it has formed in the third Congress. Fourth important, that is in 1924, Cambridge, that is in UK, it led to the formation of ISTA. Where is the headquarters of ISTA? From where certification originated? It has, according to this, the Canada, in Canada, the first seed certification has been originated is Canada. Okay. So tell me, where is the headquarters of ISTA? Is it Zurich, Switzerland? Okay. To remember other institutes also, and also the when did they found, where, and any background, the headquarters locations. And the next question is, see, we got a question here. Headquarter of National Seed Research and Training Center is located at, just simplifying it, NSRTC, located at option A, Bhopal, Bengaluru, New Delhi, and Varanasi. Right, people answered Zurich, right? So you answer for this. NSRTC. Right, it is located at Varanasi. So National Seed Research Training Institute, you just see here, is related to seed first, where it is taking some research activities and also the providing trainings through workshops and annual congresses, and also some giving seminars and all. In seed, it is also giving some making some partnerships with the private seed industries and harmonizing our seed industries, right? So remember this NSRTC, what are all the functions and where it is located? In Varanasi. Okay. When it is found? Zip 5. Okay. So the next question is, the site of double fertilization during seed development is? Options are polar nuclei, embryo, endosperm, and chalaja. Site of double fertilization. Answer this. Site of double fertilization in a seed. Okay. We know what is double fertilization, right? So this is the embryo sac. Okay. Is egg cell. This is the micropyle region. Okay, double fertilization means we are getting the two germ nucleus, where one will fuse with the egg cell, other will fuse with a polar nuclei, central nuclei. So if it only fuses with a egg cell, it will just, as usual, it will form into zygote. Is the fertilization. But since it is also fusing with the polar nuclei, we have termed it as a double fertilization, and this is called as triple fusion, right? Two nuclei fusing with a one nuclei. It's called triple fusion, and so two fertilization processes are taking place, so it is called as double fertilization. So the where exactly the site of double fertilization is polar nuclei. Understood? Farouk, it should be embryo sac, not embryo. Yeah, it is embryo sac. Okay, Nisha, will definitely do it for agronomy also. So the next question is, outer bract of a flower of grasses is sometimes referred to as flowering bloom, is called as. Here the whole question is just about the outer bract, which is present in grasses, that is Poaceae family, the part of blooms. It is called as outer bract, called as. Yes, Raju, it is embryo sac. Okay, if I mispronounce it, just correct it. It is an embryo sac. Okay. Okay, the question. Yeah, it is embryo sac. Right. So the answer. for this question? Try to answer for this. Which is the outer bract of a flower? They have given options lemma, palea, bract, and none. So in this image, you can see this spikelet, right? Where at base of the spikelet, we are having attached outer bracts, the two attached bracts at the base. So these are called as glooms. Among these glooms, the inner one is, yeah, outer one is lemma. They are giving protection to the seedlings. Okay, right. So the outer is lemma and palea is inner bract. Right, Sherin, Pratibha, Akash, Farooq, right. So the answer is lemma. The next question is, in the final stage of seed development, accumulation of triglycerols and oleogens takes place in which part? Options are embryo, endosperm, chalaza, and both A and B. Final stage of seed development, accumulation of triglycerols. I'll simplify, triglycerols means fats. Okay? Answer this question. Okay. At accumulation means maturation, drying. The seed will accumulate all the materials like carbohydrates, proteins, fats, and all, so that during germination and during dormancy it can survive. So these triglycerols are fats, and what are oleosins? Oleosins are fat covering proteins which are present on the outer layers of these fats or oleosins. So in dicots, where you find fats or oils? In majorly in embryo. In monocots, it is endosperm, right? But I'll tell you some exceptions. The castor, majorly the oil is present in endosperm, and maize, the monocot, where the oil is present in germ. Germ means embryo. So if they will specify, if they ask specifically, is it in dicot or monocot, you just see for it. If they ask it generally, so it is both A and B. Because it dicots it is embryo, in monocots it is endosperm. Try to remember the exception things. Okay. Both A and B, right, Raju, Pratibha? Okay, if it is monocots, it is endosperm. That is right. Right. The next question is, a type of ovule in which the ovule is erect with micropyle and funiculus lying on the same axis is known as Amphilotropous, asteroid, Orthotropous, and none. Answer for this. You might be seeing the types of ovules. There are many classifications or many bases for the classification, but it is based on the axis of micropyle, chalaza, and hilum. How they aligned, that is based on the type of ovules we have, right? Orthotropous, Anatropous, and all. Answer for this. Polygonaceae family, right? But what it is called as? So just see here, we have a type of ovules based on what? Relationship of micropyle, chalaza, and hilum. If you see here, the first type is orthotropous, right? You see this? We have discussed this, right? Which is connected, that is funic. And this is ovule. Here, ovule is straight, right? So that the chalaza and the hilum, all the integuments, it will merge here. Okay, and here this is the micropyle region. So ovule, if ovule is straight and chalaza and all the integuments are merging, it is orthotropous. And we have anatropous. The same funicle is there, but the ovule is inverted. See here, right? The ovule is completely inverted. So that where we have this attachment hilum, is not merging here because integuments are here. Micropyle and hilum are very close here. But here the hilum and integuments are very close and they are getting merged. But here micropyle and hilum are very close. Anatropous. The majority of the angiosperm families will display this type of ovule development. Okay, remember that. And we have amphitropous. By name itself indicates it is slightly curved. See, it curved in such a way that the embryo sac inside itself is also getting curved. And this shape is called horseshoe. Remember this. In amphitropous, we are getting a horseshoe type. And in same way, we have Camphilotropous, where it is curved, but it is not curved as much as Amphitropous. So that is Camphilotropous. And Circinotropous. Circin means circle. The funicle is too long in such a way that it is completely covering the ovule. So it is called Circinotropous. So these are all the families in which type we are in which families we are getting which type of ovules and all. So majorly Anatropous. Remember, okay? You will get this note. Don't worry. So the next question is growing point of embryo which gives rise to shoot or above ground part are referred as. It is also very basic question. Answer this. The growing point of embryo which give rise to shoot or embryo. Shoot or above ground part. Okay, we know till now we have discussed everything, right? Like embryo, endosperm, how it works, the germination types and all. So if we see this seed germination is a seed, the next radical will be protruded. From here you are getting the hypocotyle first, that is, so hypocotyle will give rise to radical or root, and the next you are getting cotyledons, that is seed leaves, we can call it as. And above cotyledons you are these are the cotyledons. Above cotyledons you are having the epicotyle which will give rise to first true leaves, that is shoot. Okay, so remember hypocotyle will give rise to root and epicotyle will give rise to shoot. Okay, okay, sorry for the interruption, there was bit a technical glitch. Okay, understood, I think. And just remember, in case of monocots, we have a coleorhiza which will cover the rhiza means root, and we also have a one sheath which is covering shoot, that is coleoptile. Okay, that's it. Epicotyl, right. It is epicotyl, right answer. And the next question is transient seed has a viability period about. Okay, 6 months, 18 months, 1 year, and 5 year. Transient seeds. Try to answer this. This is related to seed dormancy and germination, that unit. Okay. Transient seed has a viability period about how many days? Months, 18 months, 1 year, or 5 year? Right? Is for 1 year. First of all, tell me what is transient seed. We have soil seed bank, right? Where we are having seeds stored in the soil or which are present in a soil. They may be present for persistently for many years, like perennials, or they may present for a short duration, so that whenever they'll get a favorable condition, they may get germinated. Those seeds which are having a short duration in a soil seed bank, they are called as transient seed. Okay, so short duration means for one season usually, or 1 year, so less than or equal, usually in particular 9 months to 1 year we call it. The duration for transients, right? The next question is chemical which is used for testing the viability in pollen grains. So the options are like this: potassium iodide, sulfuric acid, iodine, and boron. Answer this. Chemical which is used for testing the viability of pollen grains. Which chemical we are using? So it is also a direct question where we are using iodine Ki solution, which 1 gram of iodine and 2 gram of potassium iodide we are taking to stain. What it will stain? Which part of pollen grain it will stain? It will stain starch content. Okay, if it is having a starch content, it will get stained. If there is no starch, there will the strain may be fainted strain or any no strain at all. Okay. Just remember potassium iodide is based on starch staining. And other methods we have like fluorescence diacetate also, and TTC, that is tetrazoleum triphenyl chloride also we can use, whatever we are using in TTZ test. And also we are having very basic dye, that is acetocarbon dye test. Okay. And next question is the stage of cotyledon development which provide nutrient to the embryo. We are having, there are many stages in embryo development, right? Basic zygote cell to till mature embryo or mature cotyledon. In between, we have stages like globular, heart shape, torpedo, and this is embryo. Okay, right. So if we see the globular one, where actually from here till here we have a two-celled, four-celled, eight-celled, then 16-celled, then we will get a globular, which is a very early stage of development. So just at 16 cell, the protoderm will be accumulated here, and other meristems, okay, other meristematic tissues start depositing at globular stage. But when we come to heart stage, during this is the stage where it will provide a nutrient. Okay? It is accumulated nutrients like carbohydrates and all. So just remember the heart stage. Okay? Will provide a nutrient. And what happens in a torpedo? The suspensor, whatever we are having, through programmed cell death, it will be dissociated. Okay? Got it? Right, Farooq? This heart shape. The next question is the after imbibition and radical protrusion during germination is termed as. Okay? This phase is termed as. Options are initial phase, lag phase, log phase, and adult. Answer this. This is also seed physiological aspect. The question is after imbibition and radical protrusion. That phase is termed as. I'll give a hint also. It is a it will come in a triphasic imbibition uptake or triphasic germination. Okay. Let's get into some concept where I told it is having a triphase, means three phases. One, phase two, phase three. But when it comes to imbibition, what happens in phase one? There is a rapid uptake of water from a seed, right? But what happens in phase two? Since it is already saturated in phase one, for activating some hormones, for some growth, for activating some embryo growth, and for mobilizing the reserves, it needs little amount of water. So it slowly takes a water. Slowly means it is a lag. And at the end of this period, radical protrusion starts. Don't think that radical protrusion will happen in phase. It is already started at the end of the phase 2 itself. So, radical protrusion. In phase 3, actual germination. So, remember the water where in which are all the phases seed is uptaking water in phase 1 and in phase 2, both, right? So, these both phases are imbibitional. Don't confuse, only the phase 1 is imbibition. These both are imbibition phase, and this is where actual germination takes place. So the dormant seeds we are seeing now, when it is not get germinated means those seeds which are not germinating will not enter this phase 3 itself. But all other seeds, whatever it may be, dormant, non-dormant, all other will go through both of these phases. Okay, that is why we are doing priming here. Okay, it will stop the radical protrusion here. So it will the phase 2 will be prolonged in case of priming. So this is the principle behind the priming. Hope you understood this. So let's come to question now. They have asked after imbibition and radical protrusion during germination. So where actually started radical protrusion? Here, right? After imbibition, here. So right. The next question is isolation distance is modified based on number of border rows. In which crop? Maize, rice, wheat, pearl millet. When it comes to seed production aspect, maintaining quality is very important. For that, we are following so many standards, right? Field standards, seed standards. One of the field standard, which is very important prerequisite, is isolation distance. They are asking a question, in which crop we can modify the isolation distance based on the number of border rows. Okay, right, is in maize. Okay. In maize, we can go for a border, modify the isolation distance based on the borders. In rice and wheat, since they are very self-pollinated, highly self-pollinated. In pearl millet, you can ask why not pearl millet? Why only maize? If you see the pollen load or type of pollen in maize and pearl millet, in maize it is bit heavy. So it will travel for a very shorter distance. So the foreign pollen, whatever the contaminated one from the other fields, can be stopped by planting a border rose, since it is having a very short distance travel. In case of pearl millet, it is a light pollen. It can travel up to the longer distance. That is why we have fixed the isolation distance in pearl millet, that is almost 1000 meter, right? In foundation seeds. Got it? So we are having a one more concept where the modification of isolation distance is based on the differential blooming dates. The name itself indicates difference in the bloom, that is flowering. It is also done in only maize. What is this differential blooming date? Do you have a field here? How many plants? Then we can follow this to modify the isolation. When your plants, in your plants, the 5% female plant, if when the 5% female plants have silk emergence, at the same time 0.05%, 0.05% tassel should not have, okay, not have shed the pollen. Okay. In your field, if the female plants are showing the 5% silk emergence, if there is no 0.05% of shedded or shed pollen or shedding pollen at that situation, we can go for modifying the isolation distance based on the differential blooming dates. Okay, just remember 5% in female, 0.05% in male. That is tassel. Understood? So the next question is seed production using polyploid. It is very factual question and direct question. In which crop we are using a seed production through polyploid method? Wheat, banana, soybean and sugarcane. You can see here in wheat, we are normally going for sexually producing seeds, right? We have exploited a hexaploid here. Naturally, it is hexaploid only, whatever bread wheat we are using. And in banana, we are going for a sterile seeds, that is right, but it is usually through vegetative propagants, that is suckers. And in soybean also, it is a diploid. We are going for the conventional methods. And in sugarcane, which is a polyploid, also that to aneuploid. So we have exploited the significance of polyploid seed production of sugar canes. Okay. So the next question is example for aloe and autogamous flower crop. Aloe and autogamous, rice, wheat, cotton, maize. Tell me what is the common name for allo and autogamous. Don't make it complex. Okay. Allo and autogamous means often cross-pollinated. So you just answer me now. Often cross-pollinated means what? So what? Which self-pollination and cross-pollination both will occur, but more than 33% of cross-pollination will occur. Rice, wheat are highly self-pollinated, just 3 to 5% cross-pollination is seen. And in maize is highly cross-pollinated because of protandry. Okay? Right. Cotton it is. And the next question is, seedless grapes are having the endosperm. The ploidy level of endosperm in case of seedless grapes. Haploid, diploid, triploid and polyploid. Answer for this. It is also a bit conceptual. Try to answer this. Seedless grapes. Okay. I'll try to explain one concept in seedless grapes. It is a stenospermocarp. Okay. What exactly means stenospermocarp means, as usual, in this also fertilization occurs, pollination, fertilization occurs, and seed begins to develop. But in case of stenospermocarp, what happens? Embryo, whatever developed through fertilization, will abort very, very early, so that it will give some rudimentary seeds. Okay? That is what seedless grapes, right? Where we are having a rudimentary seeds. So these rudimentary, though the embryo aborts very early, before that the endosperm has been developed as usual. So the what is the ploidy level of endosperm? Triploid. So though it is a seedless grapes, don't confuse, because of stenospermocarp, it has fertilization also, but the embryo aborts early, but as usual endosperm will be developed. Okay, so the answer is triploid. And the next question is, referee testing of seed conducted at which institute? Okay, options are CSTL, IIMR, NSRTC, and BBG. Referee testing. You have to answer this question. The referee testing. First, let me tell you what is referee testing. If we got any dispute about seed testing results, whatever the results we got from a seed testing, that is state seed testing laboratories, some people may got a dispute, like whatever the testing results may not be matching up with the performance of the seed, then they'll go to court, right? During that situation, court will assign testing of those same seed samples to central seed testing laboratory. Okay, since court is referee, so it is act as a referee for state seed testing laboratory, central seed testing laboratory. Where we have central seed testing laboratory? Right. Central seed testing laboratory. Where we have central seed testing laboratory? Usually we have one at NSRT, right? That is Varanasi. Earlier it was in New Delhi, but in 2005 they have shifted to, sorry, in 2007 they have shifted to Varanasi. Other one is at CICR Nagpur. Cotton research. For BT cotton samples, CICR acting as a referee testing laboratory, and for other seeds is CST. Okay. Next question is, transition stage between the root and shoot development is referred to as. What is the transition stage between the root and shoot? Right. Okay, tell me. Tillering, CRI, cotyledon, growth stage. Let me tell you, crown root initiation stage. CRI here. Okay. Tell me, transition stage. So you just see here, there are the normal primary roots and other seminal roots. I am talking about cereal. Let assume wheat. Okay, where we have primary roots and seminal roots and all other lateral. And at 21 days after sowing, in case of wheat, there will start one adventitious. Okay? From here, growth of adventitious roots will start. These are called crown root. From here, just above this, other roots called brace roots. But this is the stage where exactly we can differentiate into shoot and root. Till here, how can you say which is the shoot and which is? So after this only, it will develop into shoot and this side will develop into root. So CRI is a transition stage. Right. And the next question is, for identification of mechanical damage in seeds, which test is conducted? Options are ferrous sulfate, blotter test, agar plate, and ferric chloride. It is very important where you are getting a fixed question from genuineness of varieties, right? Testing of genuineness, vigor test, where you have different kinds of vigor test and all. So this is also part of that. For identification of mechanical damage in seeds, which test you are going to follow? Answer this. Right. People are answering very rightly today, especially is ferric chloride. Where we are going for this? Ferric chloride, we are using a 20% solution in which basically the legume seeds. Okay, for legume seeds, if when these are dipped into 20% solution of ferric chloride, if they have any mechanical damage, they will turn into dark color. Okay, based on the dark and light color, we will differentiate into the mechanical damaged or undamaged seeds. It is very useful for farmers, right? Before sowing itself, they can just dip it and they can test for is there any damage or not. Okay, just remember the blotter and agar is seed health test. Ferrous sulfate is used for paddy. Okay, so the next question is, exhaustion test is related to. I told you just the different kinds of test you will get in questions on this. So this is also one of that exhaustion test. Options are vigor, viability, germination, and moisture. Vigor, viability, germination, moisture. The options are so confusing that you have to be clear the concept. Okay, let me tell you, the name itself is exhaustion, means exhausting, like you and me. So just before the exam, how we get exhausted, right? So in this test also, they are exhausting all the nutrients in such a way that they are testing the seeds up to which extent it can survive or up to which extent it can germinate. So at the end, they will check for the germination test. They'll see the percentage of germination. Based on that, they will tell the ability of seed to get germinated even though it is having a very low nutrient content. So the exhaustion test is related to germination. Okay, the next question is, produce of certified seeds once brought, they may be used for up to which years? Assume I'm asking it in a self-pollinated crops. Answer me this. Once I have got a certified seeds, for how many years I can use those in case of self-pollinated? Tell me this. Options are 2, 3, 5 and 10 years. Okay, it is based on the concept seed replacement rate. Okay, by this got the answer, I think. What is the seed replacement of self-pollinated? Is yes, right? Cross-pollinated is 2 years. Hybrids is every year. Every year we have to purchase in case of hybrids, but in cross-pollinated it is 2. In self-pollinated is 3 years because it is 1/3. I am telling based on the seed replacement rate. It is 1/3, it is off, and it is 1. Okay, 800 you will get them. Okay, so the next question is KOH bleach test for sorghum is used to test genetic purity, physical purity, moisture, and seed health. This is a direct question. In sorghum, we will go for a KOH bleach test. What is the purpose of this test? Answer this question. It's also identifying of genuineness of varieties, right? Is for genuineness means genetic purity. What exactly we are going to do in KOH bleach test? What we are finding is the tannic acid. In sorghum, we have a tannic acid, the pigment present in the test tube. In KOH bleach test, 1 is to 5% of 1% of KOH, and in 1 is to 5 ratio of these two, 1%. This is the bleach test is household bleach, that is sodium hypochlorite, 5.25%. We are making a fresh solution of this and we are dipping the seeds in this solution. And for white coat, we have white coated varieties as well as brown coated in sorghum. For white coat, we are dipping for 5 minutes. For brown, it is 10 minutes. After 5 minutes, we will drain the solution when we will rinse it and we will check for the pigment. Is there any dark brown or light brown, or if there is no any pigment, there is no any staining also. So based on that, we will identify the any admixtures between the varieties. So the next question is, what should be minimum number of plants in the field for grow out test, that is GOT. Is also genetic purity test, right? Is the only purity test we are at present following for genetic purity. It is GOT test. What are all the minimum plants in the field required to test the GOT? Options are 50, 100, 200 and 400 plants. These are in terms of plants. Don't get confused with the sample size. That is different. The submitted sample for GOT is different and minimum number of plants present is different. Right. The answer is minimum should be 400 plants. That is into. Just remember this. And the next question is, headquarters of UPOV is located at. Options are France, India, England and Switzerland. Answer this. Headquarters of UPOV. Sherin, yes, right. That is Switzerland. UPOV means what? The International Union for Protection of New Varieties of Plant. It is signed under the International Convention of Protection of Varieties in 1961. From there, it has got revised 3 times: 72, 78, 1991. Okay. Headquarters is at Geneva, Switzerland. Okay. The next question is, sample which is submitted by a farmer or seed growers is known as. Options are submitted sample, working sample, service sample and farmer sample. The sample which is submitted by farmers is called as. Answer this. This is also very basic concept from sampling. Right. Called as. You know the submitted sample. What are the sample we are submitting for testing of different tests? Submitted sample. From submitted sample, they are obtaining a working sample. It may be different for different types of tests, that is working sample. And this is a service sample, whichever submitting by farmers or any commercial seed companies or any seed growers is service sample. And we don't have any farmer sample provision. And whatever the sample which is submitting by seed certification officer, right, who is doing seed sampling and is submitting for analysis, that is called official sample. Remember this. Okay. The next question is, seed certification involves how many phases? This is also very basic question. 3, 5, 6 and 8. What is the answer? Answer this. How many phases are there in seed certification? There is 6. What are all the 6 phases? Let's see this. Receipt and scrutiny of application, where we are checking for application, the farmer's name, area, is it suitable or not, and the verification of seed source, what class of seed, from where he purchased, any tags or labels associated with those seeds. Also field inspection, where we are checking for field standards, and inspection at post harvest, that is after harvesting and during processing. We will inspect, that is a fourth phase. Seed sampling and testing includes GOT also, some seed health test also. And the final phase is, after all getting a report and all, we are labeling it, tagging it, and we are granting a certificate. These are the 6 phases. And the next question is, intercropping is allowed for certified seed production in which crops? Options are cereals, pulses, oil seeds, both B and C. Answer this. Intercropping is allowed for certified seed production in which crops? Okay. There is both B and C. That is pulses and oil seeds. Because of the tillering habit and because of the tillering habit, we are not falling in seeds. In pulses and oil seeds, they are already mentioned. It is only followed for certified seed production. That is stage, not for foundation. Okay. Rule number 1. And if you want to go for intercropping, the seed certification agency, it will prepare a list of crops that you can choose as a companion crop with the main crop, right? They'll prepare a list of such crops. Those should be different genus, preferably different duration. Growth duration should be entirely different from the main crop. Also, it should not have any common pest, common weeds, and they should not be exhaustive crops. These they should not need any much nutrients and all the minerals, right? So these are the characters or conditions to choose a companion crops. But remember, if you even though you go for intercropping of the crops, case of pulses and oil seeds, only the main crop or basic crop, no, only that will be registered for the seed production. And this companion crop will not be registered as a seed crop. Only the seed crop will be registered as a seed production under seed certificate. Okay, remember this. And the next question is, epinasty is associated with which hormone? Options are gibberellic acid, cytokinin, ethylene, and abscisic acid. Answer this. Epinasty. Epinasty condition, you heard of it, but what is the hormone responsible for this epinasty? Which hormone will cause? Answer this question. It is also very important. Right. The answer is ethylene. Ethylene hormone will cause epinasty. You can see in this picture what is epinasty. This is bending of the leaves, right? When leaves will start bend means the outgrowth or overgrowth over the leaf surface, above the leaf surface, that is growth above the is adacial will be higher than the lower surface. When you see the more growth on the upper surface of the leaves than the lower surface, the leaf will start bending. So this condition is called as epinasty. Usually we will observe in tomato. Remember this. With example, you have to remember some physiological conditions. Okay, and also auxin also responsible for epinasty. They give both auxin and ethylene. Choose both. Okay. The next question is, this is the last question of our session. Very important question. In the last part, I have already told there are 6 designated diseases in India. Just make list of those and what are all the permissible limits for those. And this related to that, try to answer this. Whole embryo count method is used for detection of which disease? Whole embryo count method. Options are loose mud of wheat, kernel bunch of wheat, rust of wheat, and kernel bunch of barley. Answer this question. Whole embryo count is used in which disease? You can easily eliminate these two because these are not designated diseases, right? 6 designated diseases are in wheat, We have three. Okay, one is loose mud, second is kernel bunt, and ear cockle. Tundu disease. In rice, we have only one, that is paddy bunt, and others is ergot. Ergot in bajra, ergot in sorghum. Remember this. These are the 6 designated diseases in India. So the answer is loose mud of wheat. Why so? The name itself indicates whole embryo count method. The seed-borne disease, especially internally seed-borne, that means it is the fungi or mycelia of fungi will present inside the seed. So detect that fungi, we have to go for the examination of embryo. That is why we will go for whole embryo count method. What exactly the procedure of this method? We will take the seed samples and we will extract the embryo by soaking the seeds in 5% NOH. Okay. We will soak the seeds. But the NOH role is it will soften the tissues like endosperm and seed coat so that embryo can be extracted. Right? For embryo extraction, we are using a 5% NOH. Then we are keeping for 20 degrees Celsius incubation. After that, we are washing it in warm water that we can see the embryos. To get it very clear or transparent, we are using a lactic acid. It will make a embryo very transparent. And to examine it very easily, we need any stain. That is, here we are using 0.2% trypan blue. The stain will be helpful for easy examination. Then after that, we are going for washing for through many sieves so that we are getting only the embryos. Then we will be getting some mounting lenses. We are examining under 25x to 50 resolution of microscope. Based on the infection, we are calculating the percentage of infection. Okay. This is the procedure for whole embryo count, where we are extracting the embryo and we are examining the embryo only particular. Okay, that is why the name. Here you can Google form. Okay, we will re-upload it again, you check it. Okay. So this is the whole embryo count, which is solely used for the loose mud of wheat because it is an internally seed-borne disease. So this is the end of our session today. Hope you got some information in today's session, and very important note I want to give is at the end of these days, since only 1 week is left for examination, only mock tests can help you to revise your study. So we are providing, the AgriAddict platform is providing a very good quality mock test where you are getting a questions with a detailed explanation for each and every question. And you can also upload your doubts regarding any questions in a mock test, and you can attend it 3 times also. So we are providing mock test also. We have given that mock test series link also in our description box. Please check for it and go for it and revise it wisely, revise it quickly, and do it well in your exam. And also let me know we have to continue for any other sessions you need for seat tech or not. Please let me know in the comments. Thank you so much for your cooperation and for your response.